Hello everyone, welcome to my channel Free Learner. Today we will learn about one basic technique of molecular biology lab, PCR, polymerase chain reaction. Poly means many, merase means formation. It is a chain reaction in which this technique to make many copies of specific DNA in vitro. In vitro means in a test tube rather than an organism. PCR is developed by Karimulix in 1980. Before this machine, scientists are using 3 water bath having 3 different temperature, and in this machine they have to transfer the PCR tubes manually at least 35 times, and each water bath had a thermometer for monitor of temperature, and Karimulix also achieved the PCR amplification. So this process only. This is the traditional PCR machine. These are the instrument and also reagent used for the PCR. These are the PCR tubes, and this is the laminar hull for preparation of master mix of PCR, and this is the PCR machine. Primers, nucleotides and DNA. It has 3 steps: denaturation, annealing and extension, and these 3 steps having 3 different temperature. Denaturation, the temperature range is 94 to 96 degree temperature, and in this step we heat the reaction strongly to separate the DNA strand, and it provides the single strand template. Here breakage of also hydrogen bond. After that we can found the single strand template. Then the annealing. Annealing, the temperature range is 40 to 60 degree temperature, and in the annealing step cooling the reaction so that the primer should be add here, and here we, after adding the primer in the complementary sequence on the DNA during the annealing step, the primer provides sites for the initiation of synthesis, and the primer length should be 15 to 25 long, not less or not very long. If this is very less or short base pair of primer, then it can bind arbitrarily, and if the primer length is so long, so it can also see very slow in synthesis. So the primer length should be 15 to 25 of sequence. Then after that the third step is the extension. In extension or elongation procedure, this is the tag polymerase, which is uses in the 3' end of the primer and starts the DNA synthesis by adding the nucleotides DNTP to the growing the DNA strand. And in this step we are using the not using the DNA polymerase, we are using the tag polymerase, because DNA polymerase is very sensitive to temperature, and tag polymerase is act as the very active in high temperature, and it is isolate from the bacteria known as Thermus aquatitus, which is very active during the high temperature and induce the denaturation of the double strand of DNA. And here after completion of one cycle we are getting 2 copies of DNA, means here we are taking 1 copies of DNA. After one completion of cycle we are getting the double number of DNA. And if there is a question how we get the final number of DNA after this much cycle, 20 cycle or 50 cycle, there is also one formula, MF equal to Mi into 2 to the power n. MF means the final number of DNA, Mi means the initial number of DNA which is always provided in the question, and n is the number of cycle, and so we can easily calculate the number of final DNA after how many cycle we can calculate it properly. This is all about the DNA PCR method or multiple copies or synthesis of DNA. This is all the steps of the polymerase chain reaction. These are the modification of PCR. These are the modification: real-time PCR, nested PCR, multiplex PCR, quantitative PCR, and also the touchdown PCR. In real-time PCR, the DNA amplification is detected in real time with the help of fluorescence reporter. The signal strength of the fluorescence reporter is directly proportional to the number of amplified DNA molecule. And in nested PCR, this is the reduce the non-specific binding of product due to amplification of unexpected primer binding sites. Multiplex PCR, the beauty of multiplex PCR is that we can amplify the multiple targets of in one single PCR experiment. It amplified many different DNA sequence simultaneously. Quantitative PCR, DNA amplification linearity to detect, characterize and quantify a known sequence in a sample. Arbitrary PCR, DNA fingerprinting technique. Based on the PCR, it uses the primer to the DNA sequence in which the chosen arbitrarily. These are the applications of PCR: detection of pathogen, diagnosis of specific mutation. PCR also used for DNA fingerprinting, detection of specific microorganism, and in parental diagnosis, and also the gene therapy we are also using the application of PCR. Then what are the chemicals used for this reaction? Taq polymerase. Taq polymerase is a heat tolerance bacterium which is isolated from the thermos aquaticus, typically used in the PCR because this is tolerant in the high temperature and it also acts in the high temperature. This is a thermostable. Then PCR primer, this is a small short sequence of nucleotide that provide a starting point of DNA synthesis. DNA template, the sample DNA that contain the target sequence. Nucleotide or DNTPs, this is the single units. These provide the energy for polymerization and also building blocks for the synthesis of DNA. These are single units of bases. Buffer system, magnesium and potassium provide the optimum condition for DNA denaturation and also renaturation. It also important for fidelity, polymerase activity and also stability. This is all about the polymerase chain reaction. Thank you for watching and thank you.